Welcome to our second video on Unit 5.3 Classification of Biodiversity in our IB Biology Topic 5 Evolution and Diversity videos. In the first video, you'll see that we covered those top points in our understandings. We did also cover the international mindedness. So in this video, we'll be rolling on to looking at eukaryotes, and we will then move on to some applications and skills. So we're going to start off looking at classifying eukaryotes, and we will look at this further towards the end of the video as well. So we will pick back up on this later on. Now, when it comes to eukaryotes, you've got to remember this is an extremely diverse area with lots and lots of complex-celled organisms ranging from single-cell to multicellular like humans. You're not expected at this stage to remember this classification here, but it just shows you the diversity of our eukaryotic domain. It's another view of that wide range of eukaryotes. And remembering us as humans, we actually are multicellular organisms, so we have a range of cells that make us up. Here again, another view of that, and this kind of really shows that if we look at the Animalia, you'll see it's multicellular, and we've got a range of different cells that make up our cells. Our Plantae and our Chromista there, they all photosynthesize, hence the fact that they are green. Okay, so looking at that a little more simply, so we've got our Eukaryata over on the right hand side here, and you can see that we've got divisions of that, which are things that would be quite common to you, such as animals, fungi, plants. But then there's also these other eukaryotes. Now, if you go back to those other more complex representations in the earlier slides, they're showing the same thing but in more technical detail, more biological detail. I quite like this pictorial representation of the eukaryota there. Okay, now in the domain of eukaryota, everything is classified in the system that we spoke of in video 5.3, video 1. And we said that classification is using this system designed by Carlos Linnaeus. Okay, so where we have our domain, which we know is Eukaryata, and then we have our kingdoms, phylums, class, order, family, genus, and species. And one of your applications is to be able to choose an animal and plant of your choice and to actually know where they sit in all of these classifications. So all of these taxa, we call them. This is an example. So our taxon being kingdom, phylum, class, order, family, genus, species, and then we've got two examples, the grey wolf and the date palm. Okay, so a plant and an animal. So before we move on to looking more at animal plants, we're just going to have a further look at this understanding of natural classification. Now, with natural classification, we're looking at where organisms share a taxa. So basically, you've got these species that have evolved from ancestral species. So if we look here, for example, we have the taxa of family here. Let's look at the genus Canis. Okay, and we've got three different families. I'm trying to use a word that's not one of our taxa. So 3 different groups, okay, in the Canis genus, the jackal, the wolf, and the dog. Now, they all have very, very similar features, but they are different animals, okay, and it's kind of shown that they've come from that common ancestral species. Now, because of the common ancestry, we can expect the members of a natural group to share many characteristics. Okay, so thinking on what I've just said there. So if we'll look at the family Canidae. Here we've got the fox, the jackal, the wolf, and the dog. Okay, and there's many characteristics that they share in that group. Even if we move down to the eukaryotes, okay, there are characteristics that are living organisms, okay. Well, everything that we're looking at is a living organism, but there are characteristics between everything in this group that are not found in the archaea or the bacteria, okay. So everything within a group shares certain characteristics. Now, due to convergent evolution and adaptive radiation, which we looked at way back in 5.1, it is not always clear which group of species do share a common ancestor. So natural classification based on visible characteristics can be problematic. Okay, so when we're looking, for example, at, say we look at family again, we could look at a dog and a fox and say, hang on, they're actually the same species because they're quite similar. But this can be, if we're looking just on those physical attributes, it can be quite problematic because they're not the same species. Okay, so new molecular methods, we're talking about our DNA profiling, can now be used and have caused significant changes to classification, because in the past it has been done very much on looks. Now we can look at DNA profiling. We can actually say, hang on, no, those two organisms are not actually related or not closely related. So when it comes to reclassification, okay, and we did talk about this in an earlier video. Okay, when we're reviewing our classification, taxonomists sometimes reclassify groups of a species when evidence shows that the previous taxon contains species that have evolved from a different ancestral species. So we've got this group in a certain taxon, and one specific species there doesn't actually match the others in terms of their DNA. So they need to be reclassified. And today we've actually reclassified many species due to this evolving technology that we've got and evolving science. A good example is our asters, which we've discussed in a former video. Okay, formerly the genus Aster, comprising of hundreds of species distributed widely across Europe, Asia, and the Americas. In recent decades, taxonomists have split the group into different species. So why did we have to change classification systems? Well, natural classification uses ancestry to group organisms together, whereas artificial classification uses arbitrary characteristics such as whether or not a plant tastes good, or whose number begins with the letter C. Let's say whose name begins with the letter C, maybe not number. As early classification systems were also based on listing organisms by alphabetical order. So they found an organism, they gave it an A, found another one, gave it a name beginning with B, C, so on. A bit like we name hurricanes nowadays. Okay, so when we compare the two, characteristics and natural classification systems, we've got morphology, DNA, diet, habitat, so scientific features as such. Whereas with the artificial classification system, you had alphabetical order, human preference for taste or smell, importance to industry, and that monetary worth as well. Okay, so you see how the natural classification is much more specific and scientific. Okay, so we move on to some of our application section now, and the first one is this classification of one plant and one animal species from domain to species level. So you need to choose one animal and one plant and be able to do this, to actually classify them the full way from domain to species level. We will do that in class. If you're watching this and you're not in my class, then maybe this is a good idea for you to practice now. Our next application is the recognition of Bryophyta, Phylloscinophyta, Conifers and Angiospermators. Okay, so we are looking at phyla within the kingdom of Plantae now, so plants. And you'll see there that the plants that we're looking at, we're just not looking at Chlorophyta, okay, but the others, we've got Bryophyta, which is non-vascular, and then we've got our vascular plants there as well. Okay, so our Bryophyta, first of all, we've said non-vascular, so it's not got vascular tissues, stems, or an internal support system. So this limits their size. They don't have a cuticle, so a cuticle is a layer, like a waxy layer on top of their palisade cells as such. So what this actually restricts the movement into and out of the cell, usually if the cuticle's there. Because there is no cuticle, movement of requirements through osmosis and diffusion go directly across the surface of the whole cell. So the stomata in a plant, that's actually breaking the cuticle as such. There's a break in it. Our Bryophytes have no true roots. They have what we call rhizoids, which anchor them into the ground and allow them to absorb water. They're very small, simple leaf-like structures. They reproduce via spores in capsules on stalks held above the plant. You can see that in that top left-hand corner there. Liverwort has what we call a thallus, but no leaves. I reckon you should pronounce this Phyllocinophyta. Okay, these are vascular. They have roots, stems, and leaves. We call these leaves fronds. They produce a woody tissue, so can grow over 5 meters in height. So basically, plants need a woody lignotite to be able to grow high. Otherwise, they'll just fall over because they don't have a spinal column like we do. Some have fibrous roots, others have rhizoids. They reproduce via spores, which form in clusters called sori or sporangia. Okay, and these are on the underside of the fronds. Remember, the fronds are the leaves. And if you look in the top left-hand corner there, you can see an example of a frond with little dots underneath, and that's those sporangia. Conifers, the conifers. These contain well-developed vascular tissue, roots, stems, which are woody, and leaves. They produce separate male and female cones. Males producing pollen, females producing ovules. They produce seeds which develop on the scales of the female cones. Their gametes do not require water for fertilization. They have tough needle-like leaves with thick cuticles and a really sunken stomata. These are adaptations for water loss and dry environments. They have no flowers or nuts or fruits, but you will know the pollen off of conifers. Okay, so there's lots of pollen comes off them, but they don't actually flower. That comes from their cones. The Angiospermata are flowering plants. These are vascular. They've got tissues, roots, stems, and leaves. The gametes do not need water for reproduction. Male and female reproductive organs are located within the flower. The male's anther produces pollen. The female ovary produces ovules. Seeds produced inside the ovaries. The ovary develops into a fruit or a nut, which aids the seed dispersal. You would have looked at flowering plants way back in the junior years of school. Our next application task is now the recognition of features of birds, mammals, amphibians, and reptiles. So you'll think we've just looked at the plants, now we're looking at the animals. Before we go on to looking at our list of plants there, we just need to have a think about the fact that the animal kingdom is actually divided into our vertebrates and our invertebrates. Okay, so our invertebrates, get it around the right way, vertebrates having a backbone, our invertebrates have no backbone, and examples there. So before we get on to our list, we're just going to have a quick look at invertebrates because it is important that you still know about these. So we have our periphery. These include our sponges. They've got different types of cell, but not organised into specialised tissues as such. There's no symmetry with these, okay, so they're quite ununiform. They're all aquatic. They may reproduce a skeleton of calcium carbonate or silicon. They pump water through pores in their body wall and filter out food. They've got no nervous system or muscular tissue. Our next invertebrate is the Nidaria. These are our sea anemones, corals, and jellyfish. Cells are organised into tissues of 2 layers. They've got specialised stinging cells called nematocytes, and these sting because they're specialised stinging cells. And they catch prey with tentacles. They have one opening for a mouth, and to eliminate waste. So food goes in and waste comes out the same opening. Okay, our Platy Helminths. So these are flatworms, include marine worms, parasitic worms, tapeworms, lovely things. They have 3 layers of cells with a body cavity, with a mouth and an anus. They have a flattened appearance, hence their common name as flatworms. They show bilateral symmetry, so that's symmetry along their central plane. We now move on to our Annelida. These are segmented worms, e.g. earthworms, leeches. Some are aquatic, others live in soil, but they do like a moist environment. They have bodies divided into segments, have a very simple gut with a mouth at one end and an anus at the other. They show again bilateral symmetry. Our mollusks. Second largest phyla with over 80,000 species. Include slugs, snails, squid and octopus. They have a mouth and they have an anus. Many produce an outer shell of calcium carbonate, which is for protection. They also have bilateral symmetry of the soft body, but not necessarily of the shell. Our Arthropods Arthropoda. The largest phylum, okay, well over 1 million known, with probably as many yet not identified. A great area to venture into for your future careers. Includes crustaceans, insects, arachnids, spiders and scorpions. All have exoskeletons, body segments and jointed appendages for walking, swimming, feeding and sensing. They have a mouth and an anus, and they have bilateral symmetry. All right, so let's go on to the vertebrates. Now, these are the things that are stated that you know, remember. The most important classes of vertebrates then are the chordata. So with the vertebrates, ready? Our important chordata or vertebrates are mammalia. These are endothermic, produce milk for young and therefore possess mammary glands. Most of them have fur. They give birth to live young. Next up, the aves, or the birds, lay eggs with shells. They have hollow bones. They have feathers. They are mostly endothermic. Next up, the reptilia, also known as reptiles, exothermic. So they're cold-blooded. Our first two were endothermic, were warm-blooded. Our reptilia, exothermic, cold-blooded. They've got scales. They lay eggs, but some give birth to live young. So it's not necessarily a defining fact of the reptiles. Our amphibia, also known to us as amphibians, they've got a permeable skin, so they need to stay in water. Many undergo a metamorphosis. Okay, you would have done that back in year 8. They're ectothermic. Okay, so they are also cold-blooded, but they need to live in water. That's what ectothermic is referring to. They lay eggs without shells. And our fish. There's 3 classes, okay? We've got the jawless fish, the cartilage fish, and the bony fish. They're also ectothermic, so they're cold-blooded, they live in water, okay? They've got fins and gills and scales. Okay, so we move on to skills now, and what you need to be able to do is construct one of these things called a dichotomous key for use in specimens. What is a dichotomous key? You would look at dichotomous keys way back in year 7. They're used to identify unknown organisms. They're based on physical observable features of the organism. They can be displayed as a tree diagram or a written key, and at each step there is a choice between 2 alternatives. Okay, so that's why they're called dichotomous. So here we've got an example of a dichotomous key where we are trying to identify microinvertebrate life in a pond. So we've got our organism and we're looking at it, and our first choice is, does it have a shell or not? So if it has a shell, we'd go to the left. If it has no shell, we'll go to the right. So you see that at each step there you've got 2 choices. Dichotomous. So dichotomous keys can be trees, like we just looked at, a tree diagram, or it can be questions like this. So we're looking at a marine species, we've got a fish. Okay, organism is motile. So does the organism move, or organism is sessile, means it doesn't move. Okay, so again, you've got 2 choices, but it's written more as a list. Alrighty, we are now at the end of section 5.3. I hope you enjoyed this video. Look forward to seeing you soon, and remember to subscribe to my YouTube channel. Thanks for watching.